Thanks for having me speak today to the group. What I thought I'd do this morning is, as I thought about a talk today, and hope that we have a couple of the fellows on today. Do we have them on, Stephanie or Kim? Are they on, the fellows?
Yes, they are.
Oh, great, wonderful. I can't see exactly who's out there, but I wanted to reflect back on my career and think about what's kept me going in research. And I've been doing research now for 40 years. So that's quite a long time. And my enthusiasm for it keeps on growing. So I guess I have a love for research, and that's wonderful. I think, lucky that I enjoy what I'm doing. And then I thought about why, especially, when people think, what type of career should I have? Should I see patients? Should I do research? Should I drop out of medicine? Or what should I do? With regard to academic medicine, and what's kept me going is that research has been very exciting, and the reason hasn't only been related to our discoveries, to our ability to help people, um, to advance understanding of disease, but I really think what's excited me the most over the years is the surprises that research has uncovered. It's the unexpected results. Now, maybe the reason that I've had unexpected results is because I think I have, luckily been blessed with a talent to see things in research, and this is something when people come to my lab or when I try to help them, mentor them, I try to help them see things with their data, to be able to recognize findings. And research, at least my career, and I emphasize that we've been able to not just see what we expected, but to actually love the unexpected results. And sometimes when you plan an experiment or you test a hypothesis, you're basically looking for the answer that you expect. And if you don't get it, you think the experiment or the study failed. But it's actually, for me, been the unexpected results, the surprises I've had, the bets that I've lost, in the lab with people and research colleagues that have been the most exciting. So I love when things don't work and they show something that wasn't expected. And I love losing bets in the lab because that's what is most exciting to me. So what I wanted to do today is walk through some of those discoveries that we've had that have been the unexpected findings. So this talk today will not be necessarily a comprehensive analysis of a particular pathway or disease, but more about the findings and how we came across them and what they mean. And of course, through this process, you will learn about the discoveries and about the science, about the disease. If you have any questions, or if you want to talk about anything related to your own personal experiences, your research or questions, please. The success of this talk wouldn't just be me rambling on for a long period of time, but for others to comment along the way. So trying to just adjust something so I could potentially see you guys, which makes it hard. I don't know why I can't see everyone also. Does anyone have suggestions for the best way to see the group when I'm speaking? Or is that too complicated when you're on Zoom?
I've never been able to figure it out.
Uh-huh.
Sometimes I get lucky.
Yeah, I know what you mean. Right now I can see you, Kim. I can see you. I guess whoever speaks.
Can? Okay.
I can see you. Yeah, your very impressive background there. It's your favorite background. Yeah. Okay.
You have to go to view, Marc. You have to go to view and change the view.
Go back.
Just back. On the top there where you have actions.
And, yeah, I see that.
And see view. And you change the view to are you on gallery? You can if you click on gallery.
See you now, Mo. Whoever speaks, I can see.
Okay. Well, yeah. So now we can see you on. Yes, there you go.
Anyhow, we're not going to bother. Whoever speaks up, I can see, but I can't see the expression of everyone else. And most of your cameras are probably off anyhow. But the more you speak, the better off it is for me and everyone else. So the first surprising finding I had in my research actually was when I was a PhD student. You could see in 1988, so quite a long time ago. And the finding is shown here in this experiment. And this was, very surprising. So I was just as a peon in the lab. And so anybody that's a student in my lab or anyone else's lab, any trainee realizes that they may feel very trivial, but, it doesn't mean that you can't have a big discovery or change your boss's mind. And as you probably many of you know, I worked for the leading researcher in inflammation at the time, who just passed away 2 weeks ago, K. Frank Austin, who was a big mentor of mine, and he told me to go work on basophils and to figure out a way to culture them into mast cells, because it was the dogma at the time was that mast cells would derive from basophils. So he said, we've made a lot of progress on mast cells, and he was the most published mast cell researcher ever in the history of science so far, K. Frank Austin. So anyhow, he told me to culture basophils, and I wasn't able to succeed with that. But along the way, I ran a control experiment, which was to get human eosinophils, isolate them from the blood, and put them in culture, because the dogma at the time in the textbooks and everyone expecting, this is why I told me you need a negative control. The negative control was the eosinophils. And so we took blood eosinophils and we put them in culture in this system with human endothelial cells. And what we found was, I came back the next day, and you have to understand that the dogma was that granulocytes do not live after go into the blood. They had a very short half-life, and then they end up in cell death. So what I found the next day when I came into the lab, instead of these cells being dead as a negative control, I found that they were alive. And you can see the plot that we generated here. When they were cultured with endothelial cells, they stayed alive, for extended periods of time. This went out for 14 days. And when I presented this at the lab meeting, he was, basically in disbelief. He told me I ran the experiment wrong, and that this was, an error, and there must have been a mix-up and things like that. So I was pretty, probably I was pretty close to believing him, because he was, the professor. But I went back and did the experiment again, and lo and behold, it repeated. So I went back to the lab meeting the following week. And I showed him the results, and he said, he shook his head, and he actually, he did more than shake his head, but I won't go into that now. In any event, several months later, I, convinced him, and we developed a new paradigm that eosinophils were long-lived cells, that there were actually soluble factors, in this case secreted from endothelial cells, that were responsible for prolonging eosinophil survival. At that time, the word apoptosis was not known. Just think about that. It was and also the factors from the endothelial cells were not known. We did subsequently derive them. In this case, it was GM-CSF, but we also identified that IL-5 and IL-3 had very similar activities. So anyhow, this was the first big discovery that we had, which was unexpected. It was completely paradigm shifting. And today, this is just standard information when you read the textbooks about eosinophils. And we were at a meeting last week where people indicated that the half-life of eosinophils, even in a GI tract, is 6 days, consistent with our findings. So anyhow, the next set of experiments that were completely unexpected was really this. Really summarized in this paper here, which we just published a couple of years ago in the Annual Review of Immunology. And this is really, we take this for granted now, but I'd been working on eosinophils for 20 years, okay, before eosinophil depleting antibodies were developed by companies, some of them done in collaboration with us at Cincinnati Children's, especially through the early pioneering work that was taking place in terms of the first in man for some of these reagents. And we really expected, and even at the meeting last week, the International Eosinophil Society meeting, there was a lot of studies showing that mouse eosinophils do a lot of things in terms of protective immunity against germs, against tumors, developmental biology and other things. So, it was expected that human eosinophil depletion would have a number of consequences. But we've found over the years, and this is still out to be further developed, but by and large, the eosinophils are really what I call a vestigial arm of the immune system in humans, because we can deplete them and we have basically no consequences. Okay, there's no infection risk, there's no tumor risk. It appears even that there's no risk to newborn babies if they're eosinophil depleted. That data is less secure. In any event, this is summarized in this article, and I encourage you to read it. But especially the clinicians know that we have a number of these antibodies now. We have patients that have drug-induced therapeutic eosinophilpenia. In fact, some of these patients have zero eosinophils, and I'm not seeing any major consequences. So this is another major surprise that's really perplexing to me, and it continues to attract attention in the researchers, and still a lot of people certainly need more and more patients and long-term follow-up to fully appreciate that eosinophils may or may not be important. But the bottom line is that despite all the findings in mice, which are often acute models of disease, developmental problems, mammary gland problems, parasite vulnerability, cancer effects, we're not seeing any large signals in humans. And that's good for us, as clinicians, because we can knock out the type 2 immune response in humans without any major consequences. And this is just a very selective precision way to knock out only eosinophils. Now, clinical benefit in certain diseases, particularly asthma, but not have consequences. So I bring this to your attention as a major surprise, and it's still one that excites me and certainly has motivated the field. Anyone that wants to add any comments about this? Anyone that wants to raise challenge to some of these ideas? I welcome them in real time right now.
I just find it remarkable that you can get 40 years of funding for a cell type that is non-essential. So I'll just leave it at that. Mic drop on that one, right?
Yeah, that's right. That's right. This is another finding that I think is really, surprising and, for us, because when we first described eosinophilic esophagitis, and we were involved in this at Cincinnati Children's, we were really part of the pioneering group that really brought this disease to the attention of the medical community. We weren't the first to describe it, but we really did participate in uncovering its significance, its epidemiology, and certainly its pathogenesis. When we first started working on this, especially Dr. Assad and I, we were convinced that skin testing was very important and that we actually generated a lot of business for the division by having patients undergo a massive panel of skin prick testing. There was actually 60 prick tests that we used to put on everyone's back when they came in with eosinophilic esophagitis. And we were struck by how many patients, nearly all the patients had positive skin tests, usually about 15 positive skin tests. We used 60 antigens. And basically what we did was we told these patients to avoid all the foods that were positive on the skin tests. And then subsequently, the CHOP group started to do patch testing of food. And they told everyone to avoid not only the prick positive foods, but also anything that was positive on the patch test. So there was actually a time we even were doing patch testing to foods. Anyhow, this was a practice for 10 years here at Cincinnati Children's, and even today, I would say probably a lot of allergists still skin tests, when patients come in with EOE, even though it's not part of the guidelines. But the findings are summarized in this study here that disprove the value of skin testing, and this was, an important study. It wasn't the most well-conducted study, but the data has been reproduced through many other means. This is a retrospective analysis done by Carol Henderson, and who was a dietitian and nutritionist that worked with us. And she just looked at our patients, and you can see there was only about 100 patients, but she looked at what happened when these patients were put on 3 different diets. Now, the GI doctors developed this thing called the 6-food elimination diet, which was basically they couldn't skin test. The allergists were doing the directed diet. That meant that we removed everything that was positive on the skin test. The GI doctors said, hey, we don't want to waste our time with skin tests. Let's just avoid the top 6 foods, okay? So that's called the 6-food elimination diet. It actually has 8 foods, but we won't get into that particularly. But the other approach, which was actually one that was in the literature actually before we started working on this, was that this disease, EOE, responded to an elemental diet. So these were the three different type of approaches. And the y-axis is the effect on the eosinophil count. And what you can see is that all three treatments resulted in a dramatic decrease in the eosinophil levels in the esophagus. This is a log scale. The elemental diet, went down to 1. The sixth food went down to like 2, and the directed diet was like 5. But either way, all of these were considered remission, which is this line over here, 15 eosinophils per high power field. So what this data showed was that the directed diets, which is what the skin tests were based on, the skin tests were leading to, had no real benefit compared to these empirical treatments, which was the sixth food or the elemental diet. And this really wasn't accepted initially because people were avidly skin testing for the patch and the prick. These findings, along with many others, really changed the paradigm, were unexpected, and has really had big impact in terms of the patients and their diagnosis and treatment protocols. So another major finding was just reported. This was another study that we actually found an unexpected result in. And this, again, may seem trivial, but it's actually not. This is done by the Seeger Group under the main leadership of Kara Clearer. And what we see here is the results that were just published a couple of months ago. And what we tested in this study was that we wanted to conduct the first randomized controlled food multi-site study for eosinophilic esophagitis. And that's what this study is. It's actually 126 patients who were randomized to either the sixth food elimination diet, which is the one that the GI doctors recommend, and the one that was shown in the prior slide and other studies to be quite effective, versus a simple diet, which is a one food elimination diet, which is a milk elimination only. And we, of course, hypothesized that the sixth food would be substantially better than the one food. But we wanted to test this and test a series of other hypotheses. And what we found in this well-powered, very carefully conducted study that took several years, with 10 sites and part of the Seeger Group, was that the one food was equally as effective as the sixth food. Okay, and we went through a bunch of studies, and endpoints and analyses to prove that. But basically, there was very little advantage of the sixth food. So right now, again, an unexpected finding that both diets work. Both diets only work in a subset of patients, about 35 to 40% of the patients, which was unexpectedly low. But importantly, the milk alone, which is a much more simple diet than sixth foods, and certainly more than directed or elemental diet, is a very reasonable approach to initially treating a patient with EOE. So this is an unexpected finding that's kind of exciting to me. Does anyone have any comments about this or anything else that's been stated so far? Some might challenge that these are not unexpected, that these are not surprises, or some may find the flaws in these studies, which I'll be happy to talk about. Every study has its limitations.
Can I make a comment?
I was expecting that from you, Amal.
Of course. It's not unexpected that Amal makes a comment. So quite a few years ago, I wrote an editorial about a paper published by the group from CHOP on the value of skin tests and patch tests in EOE, which the editorial purpose was debunking this whole idea of multiple foods and the skin test and the patch test. And it was well received by half the people and very badly received by others. But that was an example of all publications don't have to be taken at face value, but one has to dig in them and see what's valuable and what's not. Mm-hmm. So this idea was started years ago, but I'm very glad that you eventually proved it.
Yeah. I mean, a lot of clinical research, is basically starting off with things like your editorial. It's based on a series of patients or someone's far out ideas that actually can often be accepted, such as the milk, such as peanut avoidance in the first 3 years of a baby's life. Okay, that was not based on any hard data. It was clinical, editorials and decisions that led to a massive problem that certainly contributed to the peanut allergy epidemic, and that's an example of one. But basically, what I'm trying to say is that editorials are very important, and then you need to prove that and provide evidence-based medicine, which is what this study is doing, providing evidence to actually support what you wrote about in that editorial. And that's a general theme. If you pick up the New England Journal, at half the articles are not what you'd consider to be interesting or surprising, but they're very important studies that prove things that are going to impact patients with real data. And it does take a huge effort. This case is an example of deep effort supported by the NIH and involving 10 sites and probably really huge efforts. In any event,
ask a question too.
You bet.
Have you ever gone back and just looked at the milk alone patch data?
Yeah, that was part of this study, and it was completely unrelated. That was in this study, yeah. The prick and the patch and everything. It was, also part of the findings and the recommendations. So this is another major finding that we've had. Now, when we first started working on the eosinophilic esophagitis, if someone said, what caused it? Almost everyone would have said, when we first started, it was something wrong with the eosinophils. The eosinophils were, it was an eosinophilic disease. Eosinophils were activated. There was something wrong that led to their accumulation in esophagitis. But what we found, over the years is basically no support for this theory, okay? But we found multiple lines of evidence that the primary defect in EOE has nothing to do with eosinophils, but it's rather due to the epithelium. And the reasons, that the data that substantiates that is summarized in this slide. First, we know that there's a lost epithelial barrier in EOE. Second, and I'll come to the data on this, is that the EOE susceptibility loci and genes are not eosinophil genes. Third, that we've shown, particularly Narit Azuz, that a primary upstream process is an imbalance of proteases and antiproteases that are made by the epithelium. Next, we've shown that the epithelium is pro-inflammatory. And finally, we've seen that the EOE transcriptome, which is basically all the dysregulation that we see in the biopsies on a molecular level, can be reproduced or recapitulated in a simple system by adding a single cytokine in a single cell type, which is the cell is the epithelium in vitro. And so it doesn't fully reproduce the disease in vitro, but it does mimic it to a great extent. And finally, if you think about topical glucocorticoids, basically why do we use them and why is it called topical? Because we're basically bathing the epithelium. We're not hitting eosinophils directly. We're not of course we can, but the fact is the topical therapy is very effective and we can get a full reversion of the disease, just bathing the topical region of epithelium in the steroids. So this is the genetic results, and this was again unexpected findings because this is a meta-analysis of the EOE chief susceptibility genes that we've uncovered, and there's a lot going on here by Leah Kotyan and Tremarky and others. But basically, what we see is that all of the peaks that we find, and we found now some peaks that are related to the immune system, but nearly all the peaks are epithelial cell gene products, the primary susceptibility region along the whole genome. What's important here is that this is an unbiased approach. So there's no selection, there's no hypothesis. You're just going across, in this case, nearly about 10 million SNPs, genetic variants with the imputation that we do. And what we find is that the primary defect is certainly an immune hormone, but that it's coming from an epithelium. It's coming from the esophageal epithelium. The second defect that we find, the second most common, is calpain 14, which is not an eosinophil protein. It's an esophageal epithelial protein. You look here at this one, MC. MC MC is actually an epithelial cell product. Now, it is also, thought to maybe be expressed by Tregs, but that's less substantiated. But MC is an epithelial cell transcription factor. It regulates chromatin, function in epithelium. CLEX16A is an epithelial cell product. So anyhow, there's a lot that we're uncovering with the genetics, but one of the primary messages is that we're not seeing eosinophil genes, we're not seeing primary T cell genes, we're seeing primary epithelial cell genes. Now, another set of data to substantiate this is being derived from the biopsies looking at single cell analysis. This is work done by Rockman and Lim and a number of other people in the group over the last several years. And what we found is that the epithelium is very dysregulated in the disease. And what we find is that there's a number of different epithelial cells that we've identified, and there's a transition, which is basically a movement from the basal layer, which is these quiescent cells, which includes stem cells, to a proliferating population, which is the normally the basal zone hyperplasia, the hyperplastic cells that proliferate, and then there's these cells that begin to transition, and then there's the differentiated ones that migrate up towards the lumen. If you can keep your speaker off, please. But basically what we're seeing is that in the disease, there's a complete blockage of the differentiation pathway, which is shown here. We lose this population of differentiated epithelium, and it's basically being held up at the stage of transitionary to differentiated, and there's a whole dysregulation of transcription factors that are involved in this switch that are leading to the phenotype of the disease and the expression of a lot of the pro-inflammatory genes. This is another way of showing that. This is a slingshot pseudotime analysis, which is basically looking at different populations in the normal state, where you can see the movement of these populations, and this is just quantitative analysis of remission patients, where you see still a loss of the epithelium, even in remission patients, but importantly in patients with the active disease, you lose the differentiated, you see more stuck in transitioning period, and this is another way of quantitating these changes. We're seeing these peaks that normally occur at different places compared to the normal state, which is basically an earlier stage in the differentiation. So there's a number of lines of evidence to support that there's an epithelial cell mechanism for the primary processes involved. Now, another surprise in our findings, and this is really surprising initially, because we all know that proton pump inhibitors are used for GERD, reflux disease. They target the proton pump, which secretes protons into the lumen of the stomach, and those reflux, and they change the pH in the esophagus if someone has reflux disease. So PPIs, 10 years ago, PPIs were the number one revenue generating drug. There's a whole series of interesting things in terms of drugs that have transformed the industry. But there was a time when PPIs were the most profitable drugs. They're still probably generating a lot of revenue. In any event, we know that PPIs work for EOE, and when we first started, find this, it was very obvious to everyone that PPIs were blocking reflux or blocking acid production, and it was the acid reflux that was impacting the esophagus and EOE. In fact, when we first started working on EOE, when I was a resident, actually at Boston Children's, there was actually papers coming out of Boston Children's indicating that eosinophils in the esophagus was a diagnostic criteria for reflux disease. It was, a very prominent researcher there, Harlan Winter, who was publishing that, and I learned that when I was a resident, that esophageal eosinophilia was reflux disease. So when PPIs were shown to work for EOE, it was very, logical to deduce that this was due to the acid suppressive effects. In any event, this is just one set of experiments that was recently published by Marc Rockman and other people in the group that shows that PPIs are actually having anti-inflammatory effects on the esophageal epithelium, and that they actually are ligands for a receptor called the aryl receptor, which is actually a transcription factor that recognizes xenobiotic substances, basically external substances, as well as food metabolites like tryptophan metabolites. And of course, we've got people like Nurit Azuz in the division working on this, and she recently got a grant on this. But the point is that PPI's effect in EOE has nothing to do with acid suppression. It involves different mechanisms, which are summarized in this graphical abstract, which can involve actually inhibiting IL-13-mediated signaling in epithelium, including reduction of STAT6, but also another pathway that has to do with direct effects through the aryl receptor and effects on cell proliferation and gene expression mediated by AHR dependent events. So this is important because, first of all, patients can go on PPIs, about 30% of the patients respond, but also because knowing that this is a mechanism, we can also design other therapies to target these pathways through better drugs than just PPIs, but other agents that interact with the aryl receptor and mediate similar inhibitory effects. So this is a surprise that certainly is, unexpected and one that's keeps me going. Now, another set of major findings that is developing, which is completely unexpected. I mean, this is very dramatic, finding and not yet fully accepted, but I think the data is really speaking for itself. I was actually, really the first one to coin the term eosinophil GI disease, EGID, and that was based on a long review article that I wrote in JACI. And actually, the date here is wrong; it was 2004. So it was almost 20 years ago. And I, characterized these diseases. I called them EGID. I described them. It was mainly like an editorial, really, but it was about 20 pages long because there wasn't a lot of data at the time. But I kind of put together what was existing in the literature and put EGID as intermediate spectrum between IgE and non-IgE mediated diseases and talked about some elements of them. But basically, we called these diseases EGID because of the prominence of eosinophils. We have diagnosed these diseases based on eosinophil counts, and Margaret Collins, is the world expert on this, and she counted eosinophils and continues to count them, and this is very important for the FDA and diagnosis. It's all based on eosinophil levels, and a lot of the reason for that is certainly eosinophils are prominent, and they are probably the most prominent cell in the biopsy. Although I say probably because we have evidence now that there's another cell that even may be at a higher density. But anyhow, everything in these diseases is focused on eosinophils. So what we've found in the last couple of years, and these are recently published, this is a paper that we just published a couple of weeks ago. Again, Kara Klewer is spearheading this. It's a single site study. It's a phase 2 study. It took quite a long time to conduct because of these diseases being rare. And what she found was that, and I want to acknowledge Kristen, who also was, the clinical research nurse, clinical research coordinator, who did a huge amount of work to find these patients and to keep this study, very highly conducted at a very high level of regulatory conditions and high level of integrity, of course, with the patients and things like that. So, we're looking at a multi-year study with a huge effort. And what we found, and this is done in collaboration with AstraZeneca, although we wrote the protocol and led the study, they did fund it in part. What we found was, as expected, this is expected, but we needed to prove it, that benralizumab lowered the gastric eosinophils, and this is showing you the effect on eosinophils in the stomach and in the blood. Okay. Now, do you see this patient here? This is a patient that got benralizumab that didn't respond to the drug, and that's quite interesting. We still don't understand why there are non-responders. And this is a patient who got placebo, but had a complete resolution of eosinophils. It went from 200 to 0. Does anyone have any ideas about why that may have occurred? One of the fellows, why would that happen? You guys out there? Who's out there? I can't see. Which fellow wants to talk about this? Is Stephanie on? Is Stephanie on? If
I
not,
don't
Kim,
see
can you call one of the fellows to describe this?
Okay. Helen, give a guess.
I'm not a doctor, but I'll take a guess.
Oh.
Okay, Julie. You're doing well. You're saving the fellow, huh? You're trying to.
I can put myself on the states here and look like a fool, but I would say the infection went down.
Infection. Julie, what infection are you talking about? Because there's no infection here. These are patients with eosinophil gastritis, which is an allergic disease. There's no infection.
Itis is an
Now, it may be a
inflammation.
surprise. Maybe you'll find that there is an infection.
Wait a
Now, we
minute.
are working on the microbiome here, but we have no evidence, and I'd be very excited if we showed that there was an infection. We have been looking for an infectious etiology.
Now he's
since I
giving me the
started.
education.
So maybe you know there's an infection here. That would be fantastic. That would be a big discovery.
Actually, we do have an example of at least one infection in eosinophilic gastrointestinal disease. Justin Schwartz had a patient with a worm infection. So she's
okay. But I learned from medical terminology that itis is inflammation. So there's some kind of inflammation going on here. And so, like I said, I'm not a doctor, but I love learning this stuff.
Yeah. So you said, okay, Helen, what's going on here with this patient?
It looks like eosinophilic infiltration at baseline, and then it looks like that cellularity is decreased at week 12 from baseline, and it looks like there's also restoration of, like, probably not going to use the right term, but like crypt architecture essentially as well.
Okay. Well, that was these pictures down here. I was just wondering how you describe this, and you've described it.
She's saying how to get a spurious result.
Yeah,
like
you described this accurately, and that was good, because I always say, people, need to describe their data. The interpretation of the data is open for speculation, but you accurately have described what we see here, which is this line here, which is unexpected. Okay. Now,
actually, you have four patients that went down substantially.
Yeah, they did, but they didn't go to zero. The diagnostic criteria for this disease is shown here. Okay. Anyhow, is Kristen on? No.
Yes.
Kristen, then you will tell us what happened here, and it has nothing to do with Kristen's excellence.
Yep, was not mine, but it was a patient who got active drug instead of placebo. There was a mess up.
Yep. The investigational pharmacy switched the drug by mistake with placebo and the drug. Okay.
How did you figure that out?
Kristen, how did you figure it out?
I think after we unblinded, we realized it.
Yeah.
Actually, I think it happened real time. It was with another coordinator, and after the injection was done, they looked at the box and realized that box probably wasn't right. And so we learned it real time, and it wasn't till later when we unblinded that we knew, unfortunately, that patient was supposed to have received placebo.
Yeah, it could have been, but it was patient was supposed to get benralizumab, but it was 50-50. In any event,
so was it one injection? Was it
the patient got the drug instead of the placebo? The patient got the drug instead of the placebo.
So
yes, one injection.
Study,
what?
No, just one injection.
One time.
One injection.
And the study,
and it held for
what?
So the other patients were supposed to have how many injections?
It's 3 injections. I think it was actually the last. Was it the last? Does anyone remember?
Second to last.
Second to last. But anyhow, you see a couple of things. One.
The one injection made this whole difference?
Yeah.
Yeah. That shows the potency of the drug. But I really want to point out that no one's perfect. Even Cincinnati Children's Hospital, #1 in the US, makes mistakes. You can't hide mistakes. You need to show them. We need to describe them, and we described it, and the journal took it, and a very good journal, accepted the paper, and we took this into account in the analysis. There's an intent to treat, and there's a per protocol. And this patient wasn't on the protocol per se because the protocol was deviated, but the patient is data is treated in the intent to treat as if they got the placebo. So it does change the results, and that's all in the findings. But you can't ignore something, and you can't, so you have to discuss it, and you can, and everyone makes mistakes. And this study did happen through COVID, so there were actually some other deviations in the study. But the study, just because a mistake is made, if it's a clinical mistake or whatever it is, it doesn't mean, anyhow, it's just, I think it's interesting to see the variability. So you could say that it was unexpected. It was very unexpected that a patient on placebo would get the drug. I mean, I was flabbergasted that our pharmacist could make such a mistake. But anyhow, these things happen, and it was brought to their attention, and they have rectified the procedure, and presumably, there's a lower probability of this happening going forward. I point this out. Yeah.
This is, I just want to say, probably the unexpected part is not that the pharmacy made a mistake, because this has happened with us before. Not to blame the pharmacy. The pharmacy is amazing, but this is systems, and systems do break down. But the unexpected finding is that one dose caused such an improvement. So would that mean that maybe patients can get just one dose and not 3 doses?
Yeah, well, the point about this study, which was the main point, which is beginning to be revealed in the lower image, is the histopathology, was that the eosinophils were ablated in nearly all patients, which you can see over here, okay? But the histology, which does show some improvement in some patients, but by and large showed no improvement, like in this patient over here, okay, which was unaffected, okay, was basically this drug did very little to help the patients. And this is emphasized in this heat diagram, which basically takes into account a lot of different molecular changes that are associated with the disease. And one can see that the patients look like this, which is basically this pattern of genes. This controls the normal and the EOG. And then you have our patients at baseline and following placebo or benralizumab, okay? And what you can see is that all of these look exactly the same. They don't look anything like the control. The only thing that changes is the CLC, which is the eosinophil specific protein, the crystalloidal crystal, CLC, okay, or the CCR3, which is the eosinophil specific protein, the chemokine receptor 3. But the point is that the drug is not really doing anything. And this is further emphasized here. We're looking at what's amazing, what Kristen did was treat these patients for 88 weeks. I mean, because some people might say, well, we didn't treat them long enough. Well, we actually had 2 open label extensions because we saw that maybe the double blind wasn't in effect. So we kept these guys on for like another year, a year and a half. And there was basically very few patients, if any at all, that we were convinced were getting better. And this is the endoscopy, the histology related to structural features, not the eosinophil features. The symptoms, the pain and the non-pain symptoms did not show any improvement. So we conclude that eosinophils basically in this particular disease have very little role in terms of the manifestations of the disease. Now, this is supported by another very surprising finding, but it's completely supported, the DGOS study, which is the dupilumab, the BEG study, the benralizumab and eosinophilic gastritis, which is what I just showed you. This study's acronym, the study that I just showed you is called the BEG study. This study is the Messina study. Now, this is a study which was conducted by the company, AstraZeneca, with our help. We were the primary leaders of the study. And what they found, this is unpublished, but it's in the public domain, is that when you deplete eosinophils and eosinophilic esophagitis, which is shown here, this is the eosinophil levels before and after, after the drug. Okay, this is the what is this? Oh, this is the percent reduction. You can see eosinophil levels go down 87% with the drug, and they go down 6% with the placebo. So there's a huge decrease in eosinophils in the esophagus. Yet the symptoms, the DSG, which is the dysphagia symptoms questionnaire score, was unaffected. If anything, it was actually improved more in the placebo than removing the eosinophils. So this data, which is actually a much larger study, this is about 50 sites across multiple countries, is consistent with our new information now that basically eosinophils have nothing to do with the clinical manifestations of eosinophil GI disease. And I think Dr. Collins will probably have a lot to say here about this. So we are currently, basically rethinking the whole diagnostic criteria for the disease and the role of the eosinophils and whether or not eosinophils should be used as outcome metrics for monitoring patients in the clinic, but also certainly in clinical trials based on these surprising findings. Now, if eosinophils are not causing the disease, what might be causing the disease? And there's a lot of answers to that question. But I just want to say that one of our findings that's becoming increasingly interesting and surprising, certainly surprising compared to when we first started working on this, calling this an eosinophilic GI disease, is that mast cells are very prominent and they're very activated, and they may be a very big driver of the disease. And this is, again, another analysis of single cell. And what you can see is single cells in the biopsies. You can see a very prominent population of mast cells, which is here. And there's a number of people in the group, especially Natalie Borgenstein, Ben Bruch, as well as Simin Zhang, that are heavily, and Pablo Abonia, but mainly Simin now and Natalie, that are heavily working on these mast cells, which are very angry, degranulating, and potentially, more important than eosinophils. Now, this is what these mast cells are doing in the esophagus. These are the single cells. This is looking at different cells that we can identify with the single cell analysis. And you can see mast cells are making huge amounts of cytokines. Of course, the proteases, histamine, and lipid mediators. They express receptors for like IL-33, of course, FC epsilon, AHR. Mast cells are very highly responsive to the HR ligands, including the PPIs, and we think that this could be very important. Even these cytokines are made at levels that approach that made by T cells, which are the Th2 effector cells that most people focus on in terms of the adaptive immune system. So there's a lot going on with the mast cells. Samin just found some surprising results yesterday. She was telling me, very excited about it. Last night when I was leaving, she grabbed me at the elevator, telling me about her surprising results related to mast cells. So you can talk to her about that. But this is actually really surprising. Natalie found that when you look at the single cell and you see patients that are in remission, okay, people that actually have zero eosinophils, okay, they actually have persistent mastocytosis. And this is showing you, we've called these cells the persistent mast cell. We have a resident mast cell. We've got a transient one that just goes up in the disease. And we have a proliferative one that's dividing in situ in the esophagus, which are these cells here. And what you can see, even in remission, we have this population of mast cells that persist. And what we've seen, and she's shown that these cells are actually poised. They actually express the proteases, the cytokines, IL-13, which we know is driving the disease because of the positive effect of dupilumab. And we think that these mast cells are very important, and we're currently testing that in humans now with a mast cell depleting antibody. So this is another set of surprising findings. Finally, I want to draw your attention to this hypothesis, which I think is very surprising. And this is a new thing that I'm trying to put forth, which is, again, is it surprising? No, not based on what I told you today, but is it surprising compared to when I was in medical school or when some of the fellows went to medical school or people that are currently taking anatomy classes today? I can pretty much tell you in an anatomy textbook today, they're not going to tell you that the esophagus is an innate immune organ. They're going to tell you it's a passage an organ that passes food. I mean, it's basically, I learned the esophagus wasn't really anything to be too focused on unless you had esophageal cancer or reflux, but it was basically a transit for food into the stomach, and that was its function. But what we've seen, and through the genetic analysis, through our work, is that the esophagus is basically an innate immune organ. And if you think about it, it makes a lot of sense, at least to me, that the esophagus is bombarded with antigens. It's bombarded with biophysical trauma, which is temperature, rough substances. There's a lot of foreign antigens and germs. Everything you put in your mouth goes to the esophagus. Now in the stomach, it's degraded. I mean, the stomach is no easy place either. But we do think of the stomach as a site for defensins, and we do know that it's more associated with with immunological reactions. And there are germs in the stomach, H. pylori and things like that. But people don't generally think of the esophagus as having a role. But it turns out that, if you look at a lot of the genes that mediate allergic disease, like filaggrin, for example, and the food allergy genes, such as the serpin genes, these actually are mainly expressed, or at least highly expressed, in the esophagus. So this is an ongoing hypothesis that we have, and I think the data is pretty substantial that the esophagus is an innate immune organ. So I'm going to close now by thanking everyone, the allergy people that I work with. This is all the consequences of a great team. Everything I showed today is because people come up with great ideas and they carry them out in ways that's just amazing to achieve findings that I've shown today. But the big question right now is this: what will surprise number 10 be? And who out there today will actually figure that out? Because that's what's exciting. What will be the next big surprise? I'd love to have a discussion about that, but I challenge any one of you to develop a theory, to see a result that's unexpected, whether it be in your patients, or whether it be in a mouse, or something else in terms of a gene or things like that. So I hope that we will soon have surprise number 10. I want to draw your attention to this website, which is also, it's not surprising per se, but it's extremely exciting to me that Julie Caldwell and John Bessie and Garrett Oswald have developed this site for our data to be readily available around the world in a way that's user-friendly, and it's getting increasingly user-friendly, versatile. It allows for sharing of unpublished data within the lab, which is very important, that we can upload data, like genomic data, and have it readily available for people inside the operation to probe and use, but also for people around the world to have a public arm of this EGID Express. And it allows, I think the latest hit is 3,000 different usage, or at least views on this site, and there's been dozens of papers published from people around the world that are using our information to advance science and help patients through EGID Express and data sharing, which provides a platform also for other labs to also use similar software that they've developed to upload information and make it available in a user-friendly manner. So thanks to all of you, and I'd be happy to chat a little bit more.
Just a quick comment, Mark. So Frank Austin was right.
Okay, yeah, he was right in a lot of ways. He lived to 95. He did a lot of things. He was right about leukotrienes, developmental leucast, and maybe he was right when he let me in his lab. I don't know. But okay,
No,
yeah,
he was
what's your
right.
point?
Yes.
What was your
I
point?
mean, he was right about mast cells. You're back to mast cells.
Yeah.
And not eosinophils.
Yeah, that's right.
Yeah. I'm sorry to hear that he passed away. We didn't get to notice that. He was a great man. Yeah.
Thank you. Who has an idea for surprise number 10?
I have no clue.
You do. You hypothesize that there's an infection that causes eosinophil gastritis, which would be great. You know, we've been looking for one.
Give me a couple more meetings and then I'll come up
Uh-huh.
with number 10.
Yep.
Thank you. This is very nice.
Non-pasteurized milk.
What about it?
Put the microbes back in. See what happens.
Mhm. Well, that's Platts-Mills theory. You know, it's the pasteurization that's messed us all up. But you know, I don't
People will
agree
get
with most of-
TB.
I don't agree with most of his things, but you know, he's right about the meat allergy tick story, probably.
People will get TB, Kim. And pasteurized milk causes TB.
Yeah, but you won't have EOE.
Well, what's worse?
Anyhow, I see some nice faces now that my screen isn't being shared, and glad I could entertain you. Anybody else want to bring anything up before we adjourn and move on? Okay, well, have a nice day. Bye-bye.
Thank you so much.